Welcome to this video in the Blueprint of Life topic. This video is going to be looking at the dot point, outline the processes used to produce transgenic species, and include examples of this process and reasons for its use. Biotechnology is the use of biological processes by industry or agriculture to change organisms to produce useful products or provide a range of services. Biotechnology has been used for thousands of years, for example, to bake bread, brew beer, make cheese, and breed cattle with specific characteristics. Genetic engineering is a technique of modern biotechnology where genetic material is manipulated to alter the characteristics of an organism. So, one of these particular types of biotechnology is referred to as transgenics. So when we look at the word and we can break it up into its two components, trans means different, and genics obviously refers to genetics or genes. A transgenic species is a species that has genes injected from another species into its genetic code. To produce a transgenic species, the first step is to identify the gene that needs to be inserted from the other species into the species that we want to change or improve. Often the gene for the required characteristic is injected into the nuclei of egg cells or the early embryo to become part of the DNA. So, for example, human growth hormone has been inserted into a range of different fish, such as the salmon, in order to produce larger sizes. Once the gene has been identified, it needs to be cut out of its original DNA strand using special enzymes called restriction enzymes or restriction endonucleases. Endonucleases. There are more than 800 different types of restriction enzymes, each that are able to cut a specific point in a sequence of nucleotides, and they form sticky ends. What we mean by this is then when we put the new DNA into the DNA of the other organism, the sticky ends then help other points where the DNA needs to combine. The next step in the process is annealing, when the matching sticky ends of the two cut strands of DNA are mixed together and connect. Sealing enzymes, such as DNA ligases, are added to the annealed DNA to help seal and strengthen the bond formed between the new piece of inserted DNA and the original strand of DNA. A large amount of genetic engineering has involved bacteria. Bacteria do not have a nucleus, and their DNA is a small circular plasmid that can be easily moved from one cell to another. Plasmids are used as vectors to transfer transgenes into bacteria. For example, the human gene to produce insulin has been inserted into the plasmid of bacteria so that these bacteria produce insulin. The insulin can then be extracted, processed, and used for people that have diabetes. There are several reasons for the introduction of transgenic species. Many transgenic species have been introduced to assist agricultural productivity. Examples include inserting a cloned growth factor into sheep and pigs so they grow faster, and tomatoes being modified to switch off a gene that makes them go soft on ripening. Transgenic species can also provide more food as the Earth's population begins to increase. And genetic engineering techniques are also used in the treatment of diseases, supplying designer proteins for human use, as we mentioned earlier, such as the production of human insulin by bacteria. So, a couple of specific examples of transgenic organisms. Sorry, that image has come up. I've obviously got my transition slightly out of order. So, insulin is important for people who have type 1 diabetes. If we have bacteria that make us insulin, we can use the insulin from the bacteria and people can inject it, helping us to then therefore reduce the cost of creating insulin. So, here we can see the steps in the process of creating insulin using bacteria. So, the necessary information on the gene is removed from the DNA, inserted into the plasmid of the bacteria, as we can see in this image here. So the gene is removed from the DNA of the person, the gene for creating insulin, transferred into the plasmid. So that's the genetic information of the bacteria. That is inserted into a bacterial cell, which then is obviously able to reproduce over and over and over again. As we know, bacteria reproduce very quickly. And then they are able to purify and isolate the insulin and turn it into the injectable form so that diabetics are able to use it. Another example that we have is BT cotton. So unfortunately, that diagram's come up behind there, but we've seen images of Bt cotton. So the Bt gene, or Bacillus thuringiensis, from bacteria makes the plant pest resistant. So it stops the need for pesticides in this particular crop of cotton plant. We find bacteria with the Bt gene and we isolate it, just like we did with the human insulin gene from our insulin example. We use our restriction enzymes to take out the gene, and the bacteria is then placed into a new bacteria that invades a plant cell. Once a bacterium invades the plant cell, it will combine with the DNA of the plant and cause the plant to become pest resistant. We can then create more of the same plant, and all of them will be will be resistant to that particular pest. So, the introduction of transgenic species, however, has raised several ethical issues. Ethical issues relate to environmental concerns, animal welfare, and human safety and confidentiality. One ethical issue concerns the release of transgenic species into the environment and their possible effect on other organisms. The increased ability of transgenic species to survive in particular conditions that they're created for could harm other beneficial species and therefore adversely disrupt the food web. The production of genetically modified food could also affect the health of some people, people who are allergic to different things. We're not quite sure what genes are being transferred from one species to another. So a person could be allergic to that other species and therefore unwittingly consume food that has been created using the genes of that species, and we don't actually know what adverse health effects this could have on people. So this is something that we're going to have a look at in our secondary source investigation that links with this dot point, and we'll have a look at some ethical issues around things such as as the environment and nature, financial and social justice issues, and medical and health issues, as well as lastly, animal and human rights issues. So we'll have a look at each of those 4 areas of ethics in relation to transgenic species and have a look at the argument for and against for each of those. And that brings us to the end of this video. Thank you for watching.